So a very warm welcome from me again as well, and it's my really great pleasure to introduce our first keynote, Eran Segal, who's here today to speak about personalized medicine based on deep human phenotyping. So Eran will be known to all of you. I'm confident, and he leads a large team of computational biologists at the Weizmann Institute of Science in Israel. And I really, personally speaking, I think he stands out not just in terms of the amazing quality and impact of his work and publications, but particularly in terms of his leadership and how he's developed incredibly complex and innovative approaches to really use very detailed phenotyping to much better understand human metabolism, and not really just stop there, but really try and do the hard thing, which is to translate those findings into ways of better or more precisely targeting interventions at individuals of high risk of metabolic diseases, specifically diabetes and related disorders. And I think it's particularly important to highlight, in my view, that to enhance our understanding by really doing what he's doing and doing very large-scale, detailed, dynamic phenotyping of humans, that's the opposite of what's happening so often. There's no shortage of data. There's a lot of data opportunism, and doing what he's doing is, it ain't cheap, it ain't easy. And so thank you for doing this. I really look forward to your talk. The floor is all yours.
Okay, so thank you, Claudia, for the very kind introduction, and the other organizers for the kind invitation to speak here and tell you about our work. So as a computer scientist by training, I greatly value the value of data, and I think we've seen that mainly in the publication of the Human Genome Project in the year 2000. And if you think about what happened in the 20-something years, and I know we'll hear a lot about the human genome in this meeting, we really came a long way, and it was a major breakthrough, because we went from knowing about genetic variants involved in just a handful of diseases just before the publication of the Human Genome Project, to knowing today about hundreds, sometimes even thousands of genetic variants that are involved in virtually every single disease, now that we have millions, even tens of millions of people who have been genotyped. And not only that, but it's been shown that whenever genetic evidence is being used in the process of drug development, it greatly increases the chances of success, so it lowers the cost of drug development by about 50%. So it's done a lot. Still, the cost of drug development is in the billions, so we have to drive that further, but it's really been a breakthrough. But at the same time, we also know that genetics is just part of the whole story about everything that matters, and it doesn't take into account environmental factors, nothing about our lifestyle and nutrition and everything else that happens to us after we're born. And so for that reason, we have been working in the past several years on what we call, much like we had the Human Genome Project, the Human Phenotype Project. And here, the idea, as I'll show you in the next slide, is to really take an approach that very deeply profile individuals with the broadest possible way that a large set of people has been phenotyped. And the idea and the hope is that with this type of data, we'll be able to really enhance our ability to predict disease ahead of time and therefore intervene earlier, develop better approaches for diagnosing disease ahead of time, for preventing disease, and then dramatically lowering the cost of drug development, and eventually also leading to remote, personalized, AI-based healthcare. So what is it that we do? So about 5 years ago, we set up a clinic at the Weizmann Institute, and every individual that comes to the clinic undergoes this entire battery of tests that is shown here, both clinical, physiological, and also molecular testings. So prior to arrival, they upload all of their electronic health records, so we have their medical history and blood tests. Before they come to the clinic, they fill out hundreds of questionnaires having to do with their medical history, mental health, lifestyle, current or past diagnoses and medications that they're taking. Then on site, we do many different tests, many different body measures. We do blood tests on site as well, hand grip strength, various assessments of cardiovascular health with ECG and ABI, which measures differences in blood pressure between the arm and the leg. We do voice recordings. Then we have various types of imagings that we do. We use DEXA to look at full body composition and bone density. We take high resolution images of the retina. We use ultrasound to look at fat in the liver and also at the carotids. Then when people go home, they go home with various sensors. So we use continuous glucose monitors, which for 2 weeks measure the levels of blood glucose levels every 15 minutes continuously. We use sleeping lab grade devices, but these are done from home, where people connect for 3 nights to sensors that give us 8 different channels continuously during the night, and the quality of sleep we know is a major factor of health, but has never been measured at this resolution on so many people. And of course, people then have smartwatches that we look at physical activity with. On their phone, they also log all of their diet, medications that they're taking, physical activity. And then on top of that, we biobank samples, live cells, so PBMCs, serum, and plasma. And then I think the major unique aspect of this project, in my view, is then linking all of this to all of the molecular multi-omics testing that we do. And so here we do a full human genetic sequencing. We look at the microbiome, the vaginal microbiome, the gut microbiome. We use mass spec to do metabolomics. We've started to do, but actually are looking for ways to do proteomics now. We're doing a bulk RNA sequencing, and we have a novel immunology assay that we adapted from the lab of Steve Elledge, whereby we can take hundreds of thousands of antigens, put them in phages, and then using phage display, we take the antibodies from the serum of a person, incubate them with the phages expressing the hundreds of thousands of antigens that we design, and with sequencing, we can then identify all of the antigens that are bound by a person's antibodies. So because the space of antigens that we can look at is so big, we can actually profile in one experiment the entire infectious history of a person, at least that for which a person still has antibodies for. So all of this we do on the entire cohort. We've done a lot of work to automate and significantly 5 times or 10 times lower the cost compared to list prices for these different omics using automation, robotics, and by lowering volumes at which we can do these assays. And then all of this so far we have done in Israel at the Weizmann Institute. We profiled a total of 12,000 people, where the core of the cohort is a baseline cohort of healthy individuals between the age of 40 to 70. This is a longitudinal study, so we contact participants every 1 year to see changes in their health, and every 2 years we bring them back to the clinic, and they undergo all of these tests with the exception, of course, of the genetics. And then in addition, we have about 2,000 people that have undergone a subset, sometimes the entire set of tests here, people who already have been diagnosed with various diseases. This we do in collaboration with several clinicians. As I'll show you, and the rest of the talk will be about the discoveries that we started to make with this data, but we see a lot of value in this data, and for that reason, we are now in the process of expanding this study in various different ways and dimensions. So we'd like to expand the number of people that we look at, and our next target is to reach 100,000 people that we profile, and we're in collaboration with various other research groups and organizations to do this study also in 4 additional countries, and hopefully grow more. And I think that international cohorts that look at different populations, different cultures, different genetics, different ethnicities are going to be key and very interesting. We'd also like to profile more diseases, and even though we do a very large battery of tests, we're not doing all the tests that are possible, so we also are looking to enhance the tests that we do, always taking into consideration the benefit that we get from a test versus the cost, where cost is not just money, but it's also in terms of the burden that we put on study participants. So this is the cohort that we've profiled. Obviously, it's not the only cohort. There's multiple different cohorts that have been looked at in the world. I think the main very impressive example is a cohort coming from here, from the UK, the UK Biobank, a very impressive study. 500,000 people that over 15 years ago have been profiled. It's also a longitudinal profiling, and if you look at the impact that this project has made by also making the data accessible to researchers, I think it really changed the way in which we do biomedical research. If you look at just the number of publications, if you hit UK Biobank in PubMed, you see that just last year, over 2,000 publications, and so far a total of 8,000 publications. Almost 30,000 researchers from 100 countries are accessing this data on a regular basis, so it's really made a tremendous impact. It also attracted investments by various companies to basically enhance the data set that eventually also goes to the hands of researchers. So it really had a profound impact, and by that I believe that also our human phenotype project, and we started actually to make this data accessible, free for academic researchers, is also going to make a big impact because it's very complementary to cohorts like the UK Biobank. So it's still smaller, but it's growing. It'll be one of the first ones to be international, to actually have people from multiple countries. And then there's all of these different tests, mainly the more modern high-throughput tests like the microbiome, RNA sequencing, immune profiling, untargeted metabolomics, and so on, that we are doing that are not present in the UK Biobank. And there's also a lot of interesting ideas on how to take even data from smaller cohorts that have a lot of different modalities of profiling that are not present in some other cohort, and somehow develop models that link different modalities together, and then bring these models, transfer them by transfer learning methods from machine learning to other cohorts that do not have these data. And these are some of the types of methods that we are developing in the lab. So over the past several years, the data scientists, students in my lab have been focusing on using this data to look at many different discoveries. Obviously, I won't have time to go into all of them, so what I want to do in the remainder of the talk is to give you, from a high-level view, some of the key discoveries that we've been making from this data set. And the first area I want to talk about is studies that we have been doing on the microbiome, where here the idea is to use, and in the space of the microbiome, this is the largest microbiome cohort that exists as a single cohort. And we're using that to go into analyses that are at a much deeper resolution. I'll explain that in a moment. And then trying to not stop just at the associations that we find, but really develop a program whereby we can take these discoveries and eventually go into clinical trials in humans that would test the efficacy for some of these bacteria that we are finding, because we believe that many of them are actually not just correlative, but actually causative for various indications that we are looking at. So in general, our approach is a new computational discovery platform that we developed on the microbiome, which I'll go into details in the next slide. Once we use that, we actually identify various strains that we think have therapeutic potential. And then the benefit or the advantage that we have is because we actually are, we profile and we manage this cohort, we can actually look at the strains that we identify, go back to study participants in individuals who harbor the strains that we think are therapeutic, invite them again to take another sample from them, from which we hope to isolate strains that we think have therapeutic potential, and then eventually go the path of formulating them in a GMP manner and going into clinical trials to test their efficacy. And the first two indications that we're going to be looking at to do these in are type 2 diabetes and weight loss. And so here's a high-level view of the computational platform that we developed. And so we start, as in all microbiome studies, we start by sequencing the stool samples, looking at the nucleic acids, and most of it are coming from bacterial genomes. We then have a reference set of bacterial genomes, and we map the reads that we have from each individual to these bacterial genomes. Now, at this stage, what we and others have been typically doing is to just count how many reads mapped to each bacteria, and by that map the species composition of each sample, and then do the analyses at the level of bacterial species. So we would look at presence or absence or abundance level of different bacterial species, and then associate that with multiple different traits. So what we have done here is to go into a much deeper level of resolution afforded by the large cohort that we have, and not stop just at the composition of which species of bacteria are there, but really to look at the mapping that we've done to the reference genomes and look at variation, single nucleotide polymorphism, so variation at the single nucleotide level between different individuals on specific bacterial genomes. So here's an example that illustrates this idea. So this is one bacterial species. This is by illustration, a short fragment of the genome, and you can see that we have 3 samples from 3 different individuals, and at this position we have variation. The first individual has an A, the other 2 have a T at this position, and we then associate this genetic variation with the variation that we have for the trait. In this case, we're looking at BMI, and you can see that in this illustrative example, the first individual who has an A has a higher BMI than the other 2 individuals who have a T, so this could be an association. Obviously, this is just on 3 samples, so it's an illustration. We do this on the 12,000 individuals that we have in our data set. And that allows us to basically do something similar to how you do GWAS studies in human genetics, genome-wide association studies. Here, we call it MWAS for metagenome-wide association studies. There are various technical adaptations that we need to do because unlike the human genome where we have 2 copies of each chromosome, so we can have 0, 1, or 2 for having how many of the major alleles we have. Here we can actually have multiple alleles, we can have multiple strains. There's many issues with mapping short reads to bacterial genomes, so there are actually a lot of technical adaptations that had to happen to go from GWAS to MWAS, but the principal idea is very similar. We associate genetic variation at the single nucleotide level in bacterial genomes with human traits. And the result of that, when we do this for the trait of BMI, is this Manhattan plot that all of you have seen multiple times from human genetics, except that here, instead of the x-axis being human chromosomes, the x-axis is now bacterial genomes, bacterial species. And every dot here corresponds to one SNP, one location in a particular bacterial genome, where we assess on the y-axis here the p-value of the association of genetic variation at that position with the variation of the trait, BMI in this case. And what you can see here is that the p-values, I hope you can see this, but the scale is such that we get very significant p-values, sometimes reaching 10 to the minus 60, 10 to the minus 80, and even lower p-values. So very significant associations. But I think much more interesting than the level of significance is actually the effect size. So what I'll show you in the next slide is exactly the same data, but now plotted as versus the effect size, so as a volcano plot. And so now the y-axis is the same, it's the same p-values. Every dot is still a SNP, but now the x-axis is the number of BMI points that are statistically explained by the variation in every individual SNP. And what you'll notice here is that we have many SNPs that actually have effect sizes that are still very significant and are above 1, sometimes even approaching 2 points of BMI. And 1 or 2 points of BMI can be a huge difference. It can account for 5 or 10 kilograms of body weight, depending on the height. So it's a huge effect. It's actually bigger than the largest effect sizes that have been observed by association of human genetics to individual SNPs, even though those studies were done on hundreds of thousands, sometimes with meta-analyses on even millions of individuals. So the effect sizes are huge. The reason I think it's expected is because I view the microbiome actually as an environmental factor, and we know that environmental factors have a major effect on traits like weight. This we've shown a few years ago. We had a paper where we showed, and actually in this data we see it as well. We had the genetics of the people and their microbiome, and we saw very little correspondence between the genetics and the microbiome, meaning that a very small fraction, we estimated somewhere between 2 to 6% of the variation in the microbiome is attributable to genetics, and the rest is determined by the environment. And so it's maybe then not surprising that an environmental factor like the microbiome has very strong associations to a trait like weight or BMI. What does it look like in terms of the strains? When we map these SNPs back to the strains, we can look at bacterial species like the example shown here, and where we identify 2 different strains. The strains, for simplicity, let's call them the blue strain and the green strain. And they differ from one another by having multiple SNPs that basically characterize the difference between these strains belonging to the same bacterial species. And you can see that for this species that I'm showing you here, if we only separate individuals by those who harbor the blue strain, the blue version, genetic version of the species, and compare it to individuals who harbor the green strain, we see a difference on average of 2 points of BMI, really a major effect. And not only that, but we see that if we look at these individuals 2 years later, then those individuals who harbor the blue strain associated with lower BMI, they don't have a change in weight after 2 years, whereas individuals who harbor the green strain associated with higher BMI, they actually gain on average another point of BMI after 2 years. So we believe, and not just based on this, but based also on other evidence linking the specific genes in which we see variation, like genes that break down polysaccharides, we believe that these strains, the green strain, for example, provides some propensity for obesity to individuals who harbor it. Obviously, it's not the only factor, not just this bacterial species and not just the microbiome, but it is, I believe, one of those factors that is contributing to this very complex trait. And so this is an example of one such species. Here's an example of another one. Here again, on average, about 1 point difference in BMI between 2 different strains, and also a gain of almost 1 point of BMI when we look at what happens to these individuals harboring the strain associated with higher BMI 2 years later. And so we believe that these strains actually are conferring a propensity for obesity, which is why we are now in the process of trying to isolate these blue genetic variants of this species, the blue strains, from individuals who harbor them, and then grow them together in a small consortium of bacteria and go into clinical trials and see if they can have a positive effect on weight loss. And similarly, I've shown you analyses for BMI. We're doing this for all of the different traits that we're collecting in this very large-scale human phenotype project, and we are finding many different associations for many indications. And so hopefully the idea will be to do this isolation process also in a high throughput, isolate the relevant bacteria, and then go into clinical trials to test their efficacy. We've done in a different area, we've actually done a proof of principle study to show that the microbiome actually can have a therapeutic benefit, a therapeutic effect in disease. In this case, we looked at a disease called atopic dermatitis, eczema of the skin, very prevalent disease in about 20% of the children, about 10% of adults to various degrees. And what we've done is, in this proof of principle study that we published a couple of years ago, we had only 9 participants, but every participant served as his or her own control, initially receiving dosings every 2 weeks of a microbiome transplant, a fecal microbiome transplant from a healthy donor to the patients. And each patient served as his own control by having the first 2 dosings be placebo, and then at week 4, starting with the actual fecal material transferred from the healthy donor. And so we can see, I hope you can see this, but this is one patient after week 4, after receiving only the placebo capsules. And from here, I hope from afar, but also you can find it in the paper, you can see very clearly the clinical manifestations of disease. And once we start to give the actual fecal material capsules, we see clearance, remission of disease very clearly in this individual. Here's another individual where you can clearly see manifestation of disease. And then at week 33, which is actually many weeks after week 12, which is the end of the dosings of the FMT, we still see the clinical improvement in disease. And when our clinical collaborators quantified the overall change, we see that, they scored as something called a SCORAD, a score of atopic dermatitis. And the score represents a moderate to severe degree of disease in these individuals, which doesn't change after giving the placebo. But once we start the FMT, we see on average a reduction of about 70% in clinical symptoms in this score. All of the individuals, all of these 9 actually improved to various degrees, but they all improved. A couple that had remission were given additional capsules, and they had improvement again. And so on average, a 70% improvement. And we then analyzed what happens to the microbiome of these individuals. And so here, what you're seeing is a reduction to a small number, to just 2 dimensions, reducing the microbiome by t-SNE analyses. And you can see here, these are samples from the donors. We had 3 different donors in the study, and all of their samples, as you would expect, cluster together. And they cluster away far from the samples of the patients. After we give the placebo, we don't expect and we don't see a major change in where patients are located in the space, so there's no real change in their microbiome. But once we begin to give the FMTs, we see that the microbiome of the patient undergoes a major shift and becomes not identical, but very similar, much more similar to the microbiome of his or her donor. And we see this for almost all of the patients. We saw a major change in the microbiome and their microbiome approaching something very similar to their donors, with maybe the exception of this one patient here. And so we believe that it is the FMT, the transfer of bacteria from healthy donors, that actually gave the clinical therapeutic benefit that we observed in this study. And so right now we're in the process of doing this study on atopic dermatitis on a larger cohort, and also to try and isolate the specific bacteria that we saw were transferred, and hopefully, much like I described for the other indications, hopefully then isolate bacteria that, when put together, could formulate a data-driven probiotic, if you will, that will be therapeutic for atopic dermatitis. Okay, so that's the area of the microbiome. Another very interesting test that we're doing, also it's also done as part of the UK Biobank, and there's been multiple very interesting publications on this, are the fundus images, where you can see here that by taking very high resolution images of the retina, we can see the blood vessels, we can see their curvature, their density, their number, and all of this could be quantified. And there's been multiple different publications showing that just by these retina scans of the images, you can actually tell a lot about the health status of a person. You can tell their age, their gender, their BMI, whether they smoke or not, levels of various blood tests like hemoglobin A1C, which is key for diabetes, and you can also predict disease ahead of time. So this is another data modality that is very interesting, and with the deep learning methodologies, we are now also linking this data modality to all of the other molecular markers that we are measuring. Another data modality I'd like to mention is metabolomics. In fact, from all of the analyses so far that we've done, metabolomics has the strongest association to all of the clinical traits that we're tracking. So it is in our hands, the most informative multi-omic that we are looking at. And here we took advantage of the fact that we have many different measurements on people to ask a very fundamental question, which is what actually determines levels of different metabolites. And the reason that question is important is because we know that metabolites that circulate in our bloodstream are very key to health and disease. And so if you find one of those that's relevant for some disease, the next thing you'll want to do is to modulate its level. So you need to know what factor affects it in order to modulate it. And so what we've done, and on a smaller subset of this cohort is work that we published a few years ago, is we took all of the data and we asked if we develop machine learning models that try to explain the levels of each metabolite, what is the best predictor of that metabolite? Is it microbiome, genetics, diet, and all of the other types of data that we had? And we found that we could explain levels of over 90% of the metabolites, and every metabolite was typically explained best by one of these different factors by a big margin from all of the others. So, for example, the 4 metabolites that you see here are very well predicted by gut bacteria composition and not well predicted by diet, genetics, or anything else. Here are 2 metabolites that are very well predicted by diet. One of them is obvious, a positive control. Levels of caffeine are predicted by how much coffee people drink. Okay, not surprising. But then many other many other metabolites actually have less obvious connections to diet and are yet very well explained by diet. And then there's some other metabolites that are not explained by diet or microbiome, but they have a very strong association to human genetics, so they are likely genetically determined. So by this we can begin to unravel what is the determinant of each and every one of the metabolites that we measure. And then we can take this understanding to shed light on different disease cohorts that we look at. And I want to give one example from work that we did on acute coronary syndrome and published a couple of years ago. And this is, we use this type of information to develop a new approach for personalized medicine. And so here the idea is that we take just even a single patient, a single ACS, acute coronary syndrome patient. Let's say we have a 60-year-old male who underwent a heart attack. We now go into our very large healthy cohort, and we identify 60-year-old males who are healthy, who did not have a heart attack. And they serve then as the matched reference for our 60-year-old male ACS patient. And when they serve as the healthy cohort, we can now examine every single metabolite that we measured, and we can build a distribution of what is the normal range that we expect for that metabolite, the normal range for our 60-year-old male patient. And so we can build this distribution, and then we can see where the value of this specific metabolite falls when we measure it in our ACS patient. And you can see that for this particular metabolite, it actually falls outside of the normal range. But this is for one metabolite. We can do this for all of the different thousands of different metabolites that we measure. And what we end up with is a list of all of the metabolites that deviate in our patient when we compare that patient to the correct personalized reference group for him, which are 60-year-old males who are healthy from our healthy cohort. Now that we have this list of metabolite deviations, we can cross it with the work that I just showed you before and ask the question, okay, so these, let's say 100 metabolites that we see deviate in our ACS patient, are these metabolites mainly driven or determined by diet or by genetics or by lifestyle or microbiome and so on and so forth? And by that, we can identify at least the factors that have likely been the underlying reason for the deviations and changes in the levels of metabolites for this patient compared to his healthy reference. And so when we do that, and this we published a couple of years ago, we see that even though we have two patients, say two 60-year-old males presenting at the clinic with the same clinical conditions, they both underwent a heart attack, when we analyze their metabolite deviations, we see that the drivers for these metabolite deviations, first of all, the metabolite deviations are different, but then the drivers of them may be different, where for one patient, we see that the deviations for him are deviations that are driven by diets, metabolites that are determined by diet, and for somebody else, they're genetically determined, and for somebody else, they're determined by the microbiome. And so this, in our view, also would explain why we need different treatments for different people, because the underlying cause for their metabolite deviations is very different. This same type of methodology, or looking at several different modalities, we've also been applying in other disease contexts. And another benefit exemplified here for having a very large scale cohort is that we can take a small disease cohorts, which are often much harder to collect, like a cohort of patients with multiple sclerosis or MS. Here we had close to 200 MS patients, and we could do an analysis that found matched controls for these MS patients. We find typically in our healthy cohort, we can find a 5-to-1, sometimes 10-to-1, sometimes even 15 healthy controls for each patient, and then do a very robust analysis, not just on 200 individuals, but on over 1,000 individuals to look at biomarkers that differentiate healthy people from diseased people. And that's an advantage of the healthy cohort. And we can do this on the multiple different levels that we have. And in this case, I'm showing you from this work that we've done on MS, just one of the pathways that we identified, which may actually be causative for the disease. So here we identified that MS patients have lower levels of indole propionate, which is known to protect neurons from oxidative damage. And we found this on two different levels of data, both on the metabolomic data, where we found that one of the precursors is actually lower in MS patients compared to controls, and also another precursor that is known to be generated and synthesized by gut bacteria is also lower in MS patients compared to healthy controls. And again, this is done on a robust analysis of over 1,000 individuals, where we did not have to collect anew or specifically for this study, the healthy controls, because we could go to our large-scale cohort. So this is another type of application that we have for this cohort. And here's another example showing this for inflammatory bowel disease, for IBD, comparing the microbiome of about 100 IBD patients that we identified to about 1,400 matched healthy controls, matched on gender, age, and BMI. And so here, a machine learning algorithm could almost perfectly differentiate diseased people from IBD patients from healthy controls. And we also identified several different markers on the microbiome. So we find a higher overall richness, higher diversity of bacteria in healthy controls. We find specific bacteria like E. coli that are elevated in IBD patients. And overall, we find that if you look at the amount of human reads that we have in stool samples, it's much higher in IBD patients compared to healthy controls, likely due to shedding of epithelial cells that we see from the inflammation of in IBD patients. So here again, just to exemplify the use of a large-scale healthy cohort to do an analysis on almost 1,500 people when we have just 100 IBD patients, and end up with a microbiome signature for disease. Okay, another topic I'd like to present is work that we've been doing on biological aging, and this is work where we've actually took advantage in a joint analysis of all of the different data modalities that we have. And so what we did here, I'll first show this by metabolomics. So we took here just the metabolomics data, and we tried to develop a machine learning algorithm that predicts chronological age based on the metabolomic data. And the results on held out individuals you can see here. And you can see that the predictions are fairly good. Using only the metabolomic data, we can say fairly accurately the chronological age of a person. But you can see it's only fairly accurately, because at every chronological age here, you can see that there's a large variation in the model predictions, where we have at every age many individuals that when the model looks at their metabolomic data, the model thinks that they actually look like somebody who's older. And conversely, many people where the model looks at their metabolomic data and the model thinks that their metabolomic profile resembles more somebody who's much younger. And so we asked whether these are just errors in the model, or perhaps there's clinical meaning to these model predictions. So are these individuals here at the top, are they really aging faster in terms of their metabolomics compared to individuals here at the bottom, and does that have some clinical meaning? And we found that the answer is yes, because if you compare these groups of individuals here at the top, where at every age group you take those individuals whose metabolomic profile the model predicts to be of a higher chronological age than they actually are, and you compare them to those with a younger predicted metabolomic age, you see that when you overlay independent data that was not used to develop these models, we see that these individuals here at the top are indeed in worse clinical condition. They have higher BMI, higher fat lipids, higher hemoglobin A1C, blood pressure, cholesterol, and liver enzymes. So this would be an example of just one of these, we call them biological clocks here for the metabolomic data. And much like we do this for metabolomics, we do this for 14 different body systems. We take all of the measurements that we have on this cohort and we group them into 14 different body systems, the cardiovascular system, blood lipids, immune system, and so on and so forth. And we used all of the measurements that we have on each body system to try and develop a biological clock, much like I just showed you for the metabolomic data. And so by doing that, we could actually develop a significant clock. By significant, I mean a machine learning algorithm that significantly predicts the chronological age of a person. The degree or the ability to predict chronological age varied by these 14 different clocks. So the clock based on the metabolomics was the most predictive in terms of how much it could explain chronological age. But all of the clocks were significant. And the key findings from analyzing these clocks are first, and this was somewhat surprising to me, that when we look at the pairwise correlation between all of these different clocks, we see that by and large, these clocks are not correlated to each other. They're by and large independent. So what it means is that if you're aging faster in one system of the body, it doesn't mean that you're necessarily aging faster in another system of the body. This to me was somewhat surprising, but this is what we see. The other observation was that, much like I showed you that the metabolomic clock has clinical meaning in the sense that individuals with a higher biological age are in worse clinical condition, we saw that all of these different clocks actually have clinical meaning. And not only that, but having a higher biological age for some clock doesn't just put you at higher risk for disease in general, it puts you at higher risk for a disease that relates to the body system on which the clock was developed. So for example, the clock for insulin resistance, if you have a higher biological age there, you're at higher risk of developing diabetes, and so on and so forth for some of these other clocks. The other observation was a very marked difference between males and females. And so we see here that if we look at the biological clock, this is the metabolomic clock, it sort of linearly declines with age in males. But in females, we see this abrupt change, abrupt increase just around the age of 50 to 55, which we actually link to the menopausal state of the women. So if we do an analysis that compares women of the same age but pre versus post menopausal women, we see that multiple different clocks actually exhibit a very different biological age when we compare post versus pre-menopausal women of the same chronological age. And so these various body systems are presumably affected by the menopause. The other thing that we're seeing is that if we ask about our ability to differentiate between males and females, then we always see the ability to do this differentiation, but the differences between males and females on most of the clocks actually diminish with age. So the ability to look at the data for each of these different body systems, for each of the biological clocks, and say whether that's data from a male or a female, the ability to distinguish diminishes with age. Males and females become more similar to each other on these multi-omics as we age. And then finally, when we look at metabolite patterns across age, we see various different patterns. So we see some metabolites that actually don't change so much with age, whereas other metabolites decline, other metabolites increase, sometimes in a similar fashion, sometimes in a different fashion between males and females. And then finally, I want to also touch on different work that we've done on dietary interventions, algorithm-based personalized dietary interventions. This is work that we've been doing for about 10 years, from idea to eventually clinical trials testing our approach. It started with a work that we published about 8 years ago, where we took 1,000 individuals, connected them to continuous glucose monitors, and what we observed is that different individuals have very different blood glucose responses even when they eat exactly the same food. So very reproducible within the same individual, like these two lines here coming from each individual when eating bread, but very different across individuals. We then took gut microbiome information and other clinical data and showed that we could actually quite accurately predict these personalized differences, such that we can then take pre-diabetic individuals like the one shown here with many spikes in glucose levels for 1 week of continuous glucose monitoring data, and then give this individual a diet with exactly the same amount of calories for each meal, sometimes with even pizza, ice cream, and chocolate, and still fully balance this individual's blood sugar levels. So this we showed in the original study on a short-term 2-week intervention. And what we recently completed, took us a few years, is to do a randomized clinical trial in the lab on 200 people with prediabetes, which we randomize into either a standard of care Mediterranean diet or our algorithm-based diet. And anecdotally, here's one individual with continuous glucose profiling for a full month. You can see many spikes in the pre-intervention period in the habitual diet of this participant. And then in the first month of intervention, we see a full balancing of blood sugar levels on the algorithm-based diet. And we saw major improvements in hemoglobin A1C in the algorithm-based diet as compared to the Mediterranean standard of care diet. And we saw almost double the effect on the algorithm-based diet compared to the standard of care diet. And we also saw improvements in several other metabolic parameters. So with that, I hope I showed you the benefit, the value that we can get from these large-scale and very deeply phenotyped human cohorts. And as I mentioned, given the value that we see for this, we are now in the process of expanding this study to more participants. Our next target is to reach 100,000 people. To more geographies, we are in the process of setting this up also in 4 additional countries. We also want to look at more measurements, so more phenotypes, and also to look at several diseases that we have not looked at so far. We developed all of the infrastructure in the lab, from the website in which we recruit the participants, all the IT systems and that manage participants, through the clinic that we set up with research coordinators who meet and profile and measure the participants, and then to the wet lab with automation for doing at high throughput and low cost all of the multi-omics with some proprietary assays. And then, of course, the core is data scientists, students who are analyzing the data. And we are open for collaborations to expand this study to other institutes, other organizations. So if anybody has interest, please contact me. And overall, I believe that these are the types of cohorts that will really take us to the next generation of precision medicine. And with that, I want to put the most important slide of this talk, all the people from my group who really have been working and leading these different projects. And with that, thank you for your attention.
Well, thank you so much for an amazing talk. I think, first of all, you win the prize for the best rock curve I've ever seen. So that was amazing. So I have a lot of questions, but I want to give you the opportunity. We also have online questions that Giles is going to monitor. So I will kick. There you go. Could you just hand over the... Yeah. Please introduce yourself quickly and then wait for the micro.
Oh. Jean-Michel Faustin from the University of Manchester. About your MVA study, have you also looked at the association of a specific strain or a specific SNPs with diet? Because maybe diet is the selective pressure that promotes a certain strain or a certain mutation, the diet explaining the difference.
Yeah, good question. So I didn't go into the details of how we actually do these analyses, but we set them up as a linear regression with several covariates that could account for confounders. So we use the age of the individual, even the abundance of the bacteria, and we also use covariates for diet. And they indeed eliminate some of the SNP associations that we have, but then the ones that I showed you are the ones that remain even after we account for diet with the best of our ability by the diet logging of participants.
Here, Anna.
Anna Crook from the Karolinska Institute in Stockholm. Thank you for a great talk. And actually, I had the same question as the gentleman over here. But now, if you say it isn't diet, but we're saying the microbiome is environmental, what's the environment that's giving us the wrong gut microbiome?
Yeah. Well, so first of all, I think in some cases it is the diet. Like I mentioned, some of these SNP associations actually go away when we introduce the covariates for diet. And then I think it's an open question of where we actually get our bacteria from. I personally think that it's I think of it as kind of randomly you get them from the environment. We see that some of them you get from people you live with, but that's actually a relatively small fraction. Some of them are affected by diet, and then some of them you get just from random encounters. And then each bacteria that you get, we see that actually bacterial strains are very stable across time. And so if you happen by chance to get a bacteria that gives you a propensity for obesity, then that's the one that you randomly selected from the environment. So that's what I mean by their bacteria are everywhere. and they're in the environment, and we randomly get them, or we get them non-randomly from people we live them, and some are affected by diet. So I think multiple factors affect it, but then you end up with a microbiome which is relatively stable, and that will or will not, relative to others, give you a propensity for multiple different indications.
So the data you showed were 10,000 geographically were from different countries, and so this can help explaining us adding one factor.
So all the data that I showed is actually data collected in a cohort from Israel. Yeah.
Thousands of subjects.
Yeah. The 12,000 subjects were collected in Israel. I mentioned we are now in the process of expanding the study to other countries, but we don't have that data yet. So all the data is from Israel. We have in a few of these cases, we've collaborated with other groups to validate these findings in other countries. But we really would like to set up a similar cohort with very deep profiling in other countries so that we can do joint learning from multiple countries.
Melina? No. Okay, Giles, do you have questions?
Oh, yes. So these are questions, and these are not my questions, questions from online. Someone, Oliver Robinson, asks, presumably the presence of a beneficial strain depends on the internal environment of individuals. Would probiotics be able to maintain presence of strain in absence of a beneficial environment? And do you know what these environmental factors may determine this?
Yeah. So that's a really good question. It has to do with colonization. So even if we have a beneficial strain and we try to introduce it through probiotics, will it actually persist and colonize in an individual? I think that's an open question. I think the answer is will indeed likely, like in this question, vary between people, and not all bacteria that we'll give to everybody will colonize in everybody, because we see this in current probiotics. But therefore we think that if we take multiple different strains that are beneficial and introduce them as a probiotic, then even if just a subset of them colonizes, we will see some therapeutic benefit. That's the hope.
And there's another question here from David Harley. Great talk. Have you looked at the interplay between the microbiome and the immune system to look for associations between bacterial strains and immune cell components? You also, I think, suggested that the fecal transplant led to sustained impact, or leads to sustained impact on disease. Do you see these changes sustained with the immune system?
Yeah, so another very good question. So actually in the work that I presented on, or the measurements that we do for the immune system that I mentioned, where we can profile hundreds of thousands of antigens and look at what are recognized by a person, that actually we studied in the context in comparison to the microbiome. We had several publications on this. I just didn't have time to present it here. And we do see associations between the microbiome composition and this immune system profile. The immune system profile is also very stable, like the microbiome is stable within an individual. So the answer is yes, we do see these connections, and we published on them, and we're now studying, trying to look at their clinical relevance as well.
Okay, one final question up there. Please wait for the micro.
Thank you. Maria Kordakova, Department of Multi-omics, University of Surrey. Thank you for the great talk, the first, and have you been looking at the microbiome during your dietary intervention, on taking a look on how the microbiome been changed during the dietary change? Because when we use the lower glycemic index products, we're supposed to have some changes, so this is very interesting.
Yes, so actually just a couple of months ago we published on the work I showed on the intervention of pre-diabetics. We actually looked at the microbiome composition, how it changed during the intervention, and we did see many changes. What I will comment on is that, because I mentioned before also that the microbiome is stable, so we actually see changes, and we analyze these changes, and we think that many of these changes are actually beneficial as you improve in clinical outcomes, and you can look at the paper to look at all the details. But at the same time, we see that these changes in an individual while you can identify them, they're robust, they're significant, your microbiome is still more similar to yourself than it is to other people. So the diet can have a big effect on the microbiome, but still your microbiome, or it's still going to be smaller. These changes will be smaller than the changes that you have between different individuals, unless you do something very, very drastic, like I showed, when you do an FMT or if you take antibiotics. But just by a dietary change, you can get a significant change, but you'll still be more similar to yourself than you will be to others.
So maybe I can be cheeky and ask one final question.
Yeah.
It is loaded. So I mean, I understand going to 100,000 is amazing, and I totally think your strategy of increasing the diversity of the population is great. But there's still, for the vast majority, still relatively boring, healthy individuals. So in terms of adding kind of clinical diversity, and surely if you have a translational interest, most of the value could be coming from studying the heterogeneity of many more conditions than the ones, the interesting ones you've already highlighted. So how about making this much more of a different balance in terms of a clinical study and still having enough diversity within populations?
Yeah, so we are actually right now not just increasing the number, but actually now we have a bigger focus on getting more disease individuals, exactly for the reasons you mentioned. So absolutely agree, and we are doing that. Although I will also say that we found, and this is actually interesting, that you can study disease even in healthy individuals, because disease is sometimes there's a clinical threshold at which we define, above 6.5 hemoglobin A1C, you're diabetic. But there's nothing special that happens at 6.5 compared to 6.4. So if you compare individuals that are at 6.4 to individuals who are at 4.5, you can still ask what is the difference between them, because those individuals with 6.4 will likely develop diabetes, will be diabetic soon. So in other words, the normal variation that we always see in every clinical markers that we examine on healthy individuals, you can study its association to all of the different biomarkers, and I think by that you're looking at you're also studying disease, even with the advantage of having naive individuals who are not yet taking medications. And, but we are now expanding to more diseased people.
Brilliant. Thank you so much again for a fantastic talk.
You.
Thank you.